The remote can.
Hello. All right. Here we go. All right, good morning, everyone. All right, that's great. Great to see some energy in the room this morning. I'm Oliver Glass. I'm the chair of the DYRK1A syndrome MSAB, and I want to welcome you to Dallas for our scientific roundtable. So I think before we get started, I really just want to acknowledge everyone that traveled in today. I know that they're coming from near and far, and we appreciate you coming together to discuss science around DYRK1A syndrome. We're small, but very mighty, and so we appreciate you taking the time to research, to really drive knowledge and innovation in the space. And so thank you for that. A little bit about myself. I'm an adjunct assistant professor at Duke University, and I'm also a medical director at Pfizer, none of which is related to what I'm doing here today or what I'm doing in DYRK1A syndrome. My youngest son, Ethan, was diagnosed with DYRK1A syndrome in 2020. And ever since then, we've been very much adamant on pursuing innovative therapies for those with DYRK1A syndrome, as well as developing targeted medicines, especially for him. So with that, we'll go ahead and get the scientific roundtable started. One small slight change to the agenda. So Dr. Martin Pera, who's the last presenter, he had some conflicts in travel, so he's unable to attend. So we'll have some flexibility, I think, in the day, as sort of things develop. And, if we feel like we need a break, I think we can have enough time to go ahead and insert that in there. So as you'll probably hear throughout the weekend, we have been growing as a community. I believe today we have over 900 families from 61 countries. Predominantly, you can see that most of us are coming from the United States, which probably is more representative of access to diagnosis, which is very, very difficult, often whole exome or whole genome sequencing or specific panels, and everybody's journey, having a child or somebody you care about with DYRK1A syndrome, it often can take long to achieve that diagnosis. So, hopefully we'll be moving more towards earlier diagnosis, and I'm sure, our numbers are going to be increasing, especially those that are already diagnosed with autism spectrum disorder, which probably, a good portion of them may have mutations in DYRK1A. So just a little bit about the MSAB. So the MSAB is the Medical Scientific Advisory Board. It's composed of myself, Craig, who's here, Amelie Patong, my wife, Sarah, who is also in rare disease drug development, Breji Von Bon, as well as Amy. So we've got a good broad list of expertise here, and we're again very excited to have you with us today, and looking forward to some robust discussion around some of the innovative science. So just to highlight for everyone what our key objectives are for the MSAB, first and foremost, it's to deepen the understanding of individuals affected by DYRK1A syndrome. And this is really to understand a lot of the biology and underlying genetics, as well as some of the genotype-phenotype relationship. We'll go into some of these in more detail as we kind of outline some of our grant objectives. It's also to establish a drug development toolkit and path to therapeutics. So I mentioned that we're very interested in identifying a treatment for DYRK1A syndrome, and looking to targeted genetic therapies. You'll hear from Craig and his work on N of 1 targeted therapy, and then more broadly, this strategy of upregulation of DYRK1A, either through gene expression or protein, and you'll hear more about that later as well. We're also focused on establish a research funding mechanism. Many of you are aware of our recent grant awardee, Craig, and some of the work that he's done, but we'll go over what our grant funding mechanism is and how you can apply and kind of next steps there. And also what we haven't been working towards as much, but we're going to be definitely targeting for in 2025 and in 2026 is defining centers of excellence. So centers where there's expertise in the United States in treating patients with DYRK1A syndrome, finding the clinicians who are interested in partnering, as well as those institutions. It's not easy to set up centers of excellence. Oftentimes it requires a good amount of funding, but that's definitely one of our focus as MSAB. And then lastly, and probably most importantly, educating and communicating the community, very much what you'll be presenting today. So how do we make complicated science, because let's face it, DYRK1A syndrome and things that affect the DYRK1A gene is complicated science. And it's how do we communicate that to the broader DYRK1A syndrome community in a way that allows them to really understand it better and not have it be so in the weeds of the science. So we've definitely added to our objectives, and we'll talk a little bit more about this in the next several slides, but through natural history study for deepening our understanding of DYRK1A syndrome. We'll talk about targeted therapeutics and some of the preclinical research. We'll talk about the grant funding mechanism. And then, lastly, by communicating to the community things like what we're doing today with the research roundtable, but also we'll talk about our quarterly, they're also called research roundtables, but we have these quarterly research presentations which offer an opportunity for researchers to present to patients and their families, and to really understand the science from a more holistic approach. And we'll talk a little bit about the Frontiers in Neuroscience paper. We had 4 articles that were submitted as part of this, and this features DYRK1A syndrome. So that's something that we did in the past year. You'll hear more about the Tiger study tomorrow from Emily Newhouse, so I won't really go too far into that today. It's cross-sectional data capture from a number of individuals with DYRK1A syndrome that happened years ago, is still ongoing in terms of data collection, but it's a cross-sectional study, so it's really just a single snapshot in time. And I think what we're really interested in and what we're trying to work towards, understanding what could be the best fit for us, is a natural history study that has more longitudinal data. And what do we mean by that? So, if you're 5 years old and you get a number of different surveys done, or you get a number of different assessments done, that you could potentially be getting those annually or maybe every 5 years. So you're capturing the data as an individual goes through time. And that just helps understand sort of if there's any sort of areas of regression, progression. So as you get into doing drug development and you have this data and you introduce something like a therapeutic, it just helps to understand over time what the expectation is potentially on some of these changes, or what changes are important in an individual. So maybe early on it has to do with feeding, and maybe later on it doesn't, but being able to capture that over time is really important. We're also working with Combined Brain So Kellen's here from Combined Brain, and they have a biorepository. So I don't think it was last meetup before we did a number of blood sample collections, and it wasn't all for naught. So we got blood from our kids, and we didn't waste it. So we put it to good use, and we got some great data, I think, out of it, and a potential biomarker for DYRK1A syndrome. And I'll touch upon that very briefly, but Kellen's gonna go into that much further. And then I mentioned RareX, so we're currently using the RareX platform. There's a number of different measures that are on the RareX platform that when an individual or family signs up, they can submit their genetic report to it. Granted, we have only a fraction of our community that actually have submitted the genetic report, so that's something that we're actively working on getting sort of verified in the registry. But there's 61 people that are sort of signed up in RareX, and these are a number of the domains that we're interested in terms of collecting. And I'll also talk about a disease concept model that a genetic counselor has been working with us on, as well as Kellen, and where that kind of fits into everything. But basically what we're just trying to measure some of the key areas that are important in DYRK1A syndrome. And what we'd like to do over time is get families to repeat a lot of these measures, even within the RareX platform. So again, we get that longitudinal data over time, because some of the things that were important to our children at, let's say, age 3 are going to be different when they're 13, when they're 20, when they're 25. And so this is going to be very important for us moving forward. And this is just a real quick data pull from the RareX platform. But you can see, main symptoms that are impacting our community are around cognitive impairment, constipation, feeding difficulties, most of which have autism spectrum disorder. There's coordination issues, seizures. But again, this is just kind of like a quick data pull, and we're definitely encouraging our community to continue to complete these questionnaires and surveys and be able to capture that longitudinal data over time to really identify some of the key areas that impact our kids and our family members and our loved ones. So this is something that we're really focused on. So our current sort of research areas, so we'll talk a little bit about the proteomics and disease concept model just briefly in a second. For the ICD-10 code, so we have been working for the past several months to submit to the Department of Health and Human Services. I think twice a year, they review codes that are related to, various, it could be syndromes or whatever. So we submitted a request to have a specific ICD-10 code developed for DYRK1A syndrome. And this becomes very important, if you're doing electronic health record captures, if the ICD-10 code for DYRK1A syndrome, let's say, is in that system, then you can really understand, what are the sort of treatment measures or the symptom control that's happening throughout an individual, just from like an electronic health record pull. But also this becomes very important from an insurance standpoint when you're doing reimbursement. So, for instance, like my son just had some, major dental surgery, and and the insurance companies would like to have ICD-10 codes. Like, why does your child need to have like full general anesthesia to get, their teeth pulled, when that's something that can just be done at like a doctor's, a dentist's office. So it just helps in that process, and that's really important. So this fall, we're going to hopefully, have the ICD-10 code come up and fingers crossed that it'll go forward and we'll get that approved. But that's one thing that we'd worked on. Craig's going to talk today about the ASO N of 1 project. So this was, something that we funded last year. That was our first grant that we funded. So very exciting to hear more about what Craig's working on. And then I'd also mentioned just the overall strategy for upregulation of DYRK1A. So as many of you are aware, or some of you that aren't, DYRK1A syndrome is mostly caused by a haploinsufficiency of the DYRK1A gene, meaning that you're not producing enough of ultimately the DYRK1A protein. And that's one of the major, causes of the symptoms and things that we see from DYRK1A syndrome. And there's various strategies that you can utilize to upregulate DYRK1A. I've heard from many of you an interest in sort of the gene therapy. So you can edit the DNA, you can target the RNA, you can use, stabilizing molecules and proteins. I mean, there's various strategies, but I'd like everybody to know that we are actively looking at all those approaches. And, as part of the MSAB, I think it's our job to really discern what strategies are worth supporting and moving forward, and what might have the best bang for the buck, and is going to hopefully be something that's going to come to fruition within the next couple of years. So I had mentioned briefly about the proteomics project as well as the disease concept model. Kellen's going to talk more about the proteomics project, but this really stemmed from, again, the blood samples that were collected at the family meetup, which is why this is, so important that you're all here and that, participating in research in our community. And Helen's going to talk about a potential biomarker that we found for DYRK1A syndrome called GFAP. And that's pretty exciting because I think, having a potential biomarker for our syndrome is something that can be really useful if you develop any sort of treatments and you want to see, hey, is there something that we can measure that changes biologically? Because clinically there can be a number of changes. You can increase a number of word counts, like you could be treating constipation, you can be treating all these different things, but having like sort of biological marker that you can measure is something not only important for clinical drug development, but also just something in general that's going to be helpful to understand whether you're making improvements in DYRK1A syndrome. Also, we've been working with this genetic counselor, Olivia Salverson, as well as Kellen, on a disease concept model. Many of our families participated in this through interviewing, but one of the key aspects of the disease concept model is really hearing from the clinical perspective in terms of what they think is important when assessing DYRK1A syndrome, as well as from the family members and the caregivers. So in published literature, there's a number of things that they say impact DYRK1A syndrome, but not all of that is captured in the literature. And so by having interviews with a lot of the family members, you say, hey, you know what, this is important. This is important. This is actually the most important. And when you capture that, it tells us, okay, we actually need to be putting out surveys or questionnaires or things that are validated to assess what is important in our community that isn't already being captured. And that's something that can help supplement what's already going on in the RareX registry. So when we have gaps, is what I would call it, in how we're sort of understanding and defining DYRK1A syndrome, then we're going to be able to capture that data and then hopefully, be able to build upon things that are important to the community moving forward. So just to kind of highlight again what we've been able to accomplish in the past several years, and really in the last year, we've had a dramatic and exponential increase in what the MSAB has been able to really successfully do. And that's in collaboration really with everybody. And I can't emphasize that enough. All of the researchers that are here and everything that has gone on, it's because I said yes. You're like, yeah, I want to help. And you think at the time that it's just you and like, well, what, I'm just one person, but really like the one person is what's driving science in our community. And so we can't thank you enough for that. So again, Kellen's going to talk about the biomarker project. We've got the disease concept model. Hopefully that's going to be published soon. The ICD-10 code will definitely keep you up to date on. Craig's going to talk about the ASO project, and upregulation of DYRK1A is sort of like our broader treatment strategy. It's possible, and there's definitely some preliminary data out there that it can work, and so we're definitely focusing on moving that forward. I mentioned that we have these quarterly virtual research roundtables. So Helen, who's here, participated in one. Kellen was our first, I think, presenter for the virtual roundtable. So if anybody's here, you're interested in presenting virtually, I think we're due because the last one was in April. So if you're interested in presenting to the broader community, something that's more of like, it still has, a good amount of science, but it is also distilled enough that our patient community can understand what your work is. But we're looking for presenters. So if you're interested in presenting, shoot me an email and let's make it happen. We also had a feature in Frontiers in Neuroscience on DYRK1A syndrome. So there was a call for submission of articles. So we had 3 original research articles and 1 review that were accepted in this. And this is really amazing because this, again, highlights DYRK1A syndrome in a really impactful journal. So, I encourage all of you, if you go ahead and check out some of these articles, but these were published recently in that journal. So our competitive grants program, I mentioned Craig was the first recipient of this, and this is really to focus on DYRK1A syndrome and really accelerating research in it. So these are 1-year grants up to $30,000 per participant. It is possible that funds may exceed that, but you'd have to provide some appropriate justification for that, depending on what type of project it is. And so for the process, you submit a letter of intent. This is just a really 1 page that kind of outlines what the plan is and what you plan to do. You can submit that to grants@DYRK1A.org. You can also send it to my email as well. And then once we kind of review that, then you'll get invited to submit the full-length application. And at that time, there's a bunch of instructions for what goes into the full-length application. So I encourage everybody to check that out. So these are the grant areas that we have that are of significant priority. I don't really have to touch on all of these in significant detail. But these have to focus on DYRK1A syndrome, and we're very interested in sort of some of the in vivo assessments of DYRK1A, and as well as really the novel precision medicines or targeted therapies that correct for DYRK1A haploinsufficiency. So we're very focused on those approaches, but we're also obviously interested in a number of different areas that impact our community, especially sort of gut-brain axis, impacting gut function. You saw from just that RareX poll that constipation and GI issues significantly impact our community. So Again, we're looking for innovative approaches. So we've been obviously busy on the MSAB, really kind of addressing our key objectives, and we really appreciate the partnership with you all and your focus and passion in understanding DYRK1A syndrome is commendable, and we just appreciate working with you. So I think with that, I'll say thank you. And again, just really appreciate everybody being here today. I think the idea is to really have robust discussion around, the research and things that are happening in DYRK1A syndrome research. So I encourage you all to ask questions and get to know one another. We're not a large community. But again, we're mighty. So thank you again for your attendance. And I think with that, I'll pass it along if anybody has any questions for me.